I originally was intending to give a slightly different talk, but I reverted to a talk that's slightly older because of relevance to the politics of today. But before I'd like to start, I just got this from Lorna Roll, and she, as most of you know, works at the NIH, and it said, this was an official notice from the NIH executive. Dear staff, travel, effective until further notice, in compliance with guidance from HHS, all government travel is suspended. Staff currently on official government travel can complete their trip as long as they are not presenting, but must return to the original point of departure. Future requests for any reason are not authorized. Now, I don't totally understand this, and then communications. Until further notice, all government employees must refrain from participating in any public speaking engagements, including any scientific presentations with outside organizations. So that is what your government is doing, and presumably that will only last for a short period of time before it is overturned by any number of other events. But what I would like to say is you'll see in a moment that I ended up, for a variety of strange reasons, doing work that's really relevant to climate change, which is why when President Trump pulled us out of climate change accords, et cetera, I went back to this seminar just in homage to what should be. And I'd just like to say that this project really sort of evolved from purely basic curiosity-driven science, and it sort of led us in a strange set of pathways to a whole series of questions about the effects of climate change on the invertebrate species that we study. And these are as important, I know many of you don't believe this, but it is as important to our world as the vertebrate species that are represented in this room. And these species fuel our food chain, and they are really critical to the whole life of the planet. Now, what I have learned in the process of going around and talking at meetings and visiting places, that there are a number of things about the effects of temperature specifically and climate change on humans and vertebrates that I wasn't really aware of. And I'd just like to, at the onset, just start with this rather than finish to this. First of all, many neurological disorders are very sensitive to temperature. So there are seizure disorders, such as Dravet syndrome, in which, in this case, children are incredibly temperature sensitive. Multiple sclerosis, people with MS are very temperature sensitive. The likelihood of a minor stroke turning more serious is way enhanced by temperature extremes. Things like human hyperkalemic periodic paralysis, again, is a disorder where temperature and, in this case, high potassium are linked completely in the pathology of the diseases. And almost anything, any disorder or any syndrome in which the brain is at least partially compromised, but might be perfectly normal under normal circumstances, may be exacerbated or further compromised by temperature extremes. And so I think it's really, and as we go through my talk, I think you'll understand why anything that compromises the brain will be exacerbated by, in particular, by high temperature, but also for some individuals by low temperature. And then I just came back from Marseille, where I learned something that I didn't know. I love going to places and learning something. I can still learn. And what I learned is that, it's right there, is that rodent pups, when born, are about 25 and 26 degrees, and they can't thermoregulate. And they learn, the circuit that allows them to thermoregulate develops over the next couple of weeks. And Frédéric Brachard has been working on development of motor systems in rodents, told me that as the animals mature, there are thermosensitive mechanisms that produce plateau potentials in the motor neurons that actually enhance the animal's ability to locomote. So in this case, the animals are born unable to thermal regulate to the temperature that they need in order to locomote, and that these mechanisms actually mature later in life. So I find this really, really interesting for two reasons. First of all, scientifically, it's really interesting. And second of all, I don't know why nobody told me this. And I've given this talk in one form or another in many audiences, including many, many audiences with many, many people who work on mice and rats, and no one ever told me this. And now why not? And I think that's a really interesting piece of sociology. Either you guys don't know, or you didn't think it was important. It is important, and you probably know, but maybe you didn't know. So, and the first paper published about this was done in the 1970s, just so. Okay, so before going on, I'd just like to acknowledge the wonderful people who've gone through my lab and who've contributed to this work and a lot of other work. At least two of them are in the audience, Toli and Alex, and it's really great to see you. I love seeing my ex-lab members when they've gone on to bigger and better things. And my present lab, sort of is right up there. And then I have a whole bunch of lab alumni, and many have gone into industry and government, many have gone on into academic science. And it's really, really, really one of the great pleasures to run into them afterwards and see that they still smile, right? Okay, so the topic for today is what I call degeneracy. And here I'm using the word the way Jerry Edelman used it as a geneticist, to mean multiple mechanisms that can give rise to very similar behaviors. And this can be seen in our case, both in experimental and computational work. And the reason this is important, and I'll just go back to the wild type audience that we have in this room, every person in this room has a degenerate solution to being alive. You all have respiratory centers, they're all working. And I know they're working because you're all breathing. And no one in this room thinks that your respiratory centers are identical. So that tells you there are multiple solutions to building a respiratory center, and that one of the challenges is to understand which features of that common solution, that is to say your breathing, are common, and how much variability and variance there can be in any one of the parameters in that system. I think one of the worst things we did as biologists was to learn how to calculate means. And what? It's true. You know, we like to calculate means because if you're an experimental biologist, you're very aware of all the mistakes you can make in doing an experiment. So you calculate a lot of means, and that makes you feel better. And especially if you have a small standard error, that makes you feel even better. But that is actually hiding all the degeneracy that's really important to understanding a wild-type population, or for that matter, even a genetically inbred population. So I want you to keep that in mind. And one of the things that I like to make a joke about, but I'm dead serious, is the lobsters and crabs that we study are wild-caught. They have lived in the ocean for 5 to 7 years. They're usually around 7 years old. And therefore, they're a really, really good model for you, a much better model for you in many ways than the genetically inbred animals that you study in the lab. And we can have a fight about that, but having a population that actually captures the implications of a wild type group, I think, is really important. Now, the preparation we study is the crustacean stomatogastric nervous system, and it was originally devised as an experimental preparation by Don Maynard in the late 1960s. And what he and his cohort were looking for was a preparation which could be isolated in vitro to allow detailed electrophysiological investigations of the mechanisms that gave rise to rhythmic motor patterns. That's what they were interested in at the time. But that would generate motor patterns, fictive motor patterns, that resembled very closely the motor patterns that would drive behavior in the intact animal. And so he and a lot of other people, Ted Bullock, would get on a boat and go riding around the oceans and looking for animals with particular that had particular solutions to being alive and doing something. And one of my postdocs just said to me yesterday, she said, now I understand why the STG has maintained itself as an experimental preparation for so long. And she said, I never really understood it until I started reading all about a whole lot of other circuits. And the reason is, it's a small circuit. Every neuron can be recorded from unambiguously, identified unambiguously. It's a small enough network so that you have a hope to understand its dynamics, and it's a large enough circuit or a large enough network so it has some of the richness that the networks that are doing more complicated things have. So I have studied it now since I was in graduate school. I have changed the problems with which I've used it. So I'm not doing exactly what I was doing in graduate school. You might say, thank God. But in some ways, the flavor of the early questions that motivated me then is still there. But I think, there are basically two kinds of scientists. There are scientists that move from problem to problem to problem, system to system to system, and others who just keep going, following in the path that they get taken by, in this case, by the crab and the lobster. Okay, so the isolated STG produces essentially the same motor pattern as seen in the intact animal. The top of the slide shows a crab with a hole in the carapace. The carapace has been removed, and in this case, Marie Goeritz implanted a suction electrode on a nerve called the LVN, the lateral ventricular nerve, and that's shown in that second trace. And there you can see a rhythm we call the pyloric rhythm. It's a triphasic motor pattern, and you can see it as discharges of a neuron called the LP, followed by PY, followed by PD. So it's a constrictor 1, constrictor 2, dilator, constrictor 1, constrictor 2, dilator. Now, there are 26 neurons in the crab's stomatogastric ganglion. There are 40 sets of striated muscles that move a very complicated mechanical device. So this system is more akin to your shoulder or your mouth than it is to your gut. It has actually very few similarities to your gut. It's really close to a locomotor system, except the stomach is moving inside the animal, which puts different kinds of constraints on it than if we're outside. Now, if you remove the stomach from the animal and you now place it in a silver rod-lined petri dish and put on extracellular recording electrodes, you see the kind of recordings you see on the bottom trace, which again is LP-PYPD, LP-PYPD, and you can see that rhythm looks very similar to the one above. So that was one of the major advantages of the system, that when you record the motor patterns, you're recording the output of a circuit, and that output has been very little changed by its isolation. Okay, now I want to just show you the next piece of our argument about degeneracy. And these are just recordings that Maru, who was a graduate student in my lab, who's now at the Allen Institute, where to make this slide, she just pulled random recordings from her notebooks, and recordings from 4 animals, animal 1, animal 2, animal 3, and animal 4. And you're seeing here intracellular recordings, simultaneous intracellular recordings from the LP and the PD neuron. The LP, as I said before, is one of the constrictor motor neurons. The PD is a dilator, and they fire out of phase. And one of the important things that the circuitry has to do is make sure these neurons are kept in alternation. Okay, and the LVN, that same neuron you saw before, is down below. Now, if you look at these recordings, you see obviously they're not to the millimeter identical, but they're very similar. And as I said, Mara didn't go looking for them specially. She just pulled them out more or less randomly. And so this is saying that each animal has a very stereotyped motor pattern, that when we start our experiments, we start in animals that have been isolated and dissected more or less in the same way. And this is true of every lab in the world that does this preparation. And here are 4 PD cells. And then with Dave Schultz's lab, he was an ex-postdoc of mine who's now at the University of Missouri. We've done single-cell RT-PCR on a whole series of ion channel genes. And what we did to do this in the first set of studies, we identified the neurons, recorded electrophysiologically, Jean-Marc Gaillard did voltage clamp analyses of the membrane currents in the cells. Then we pulled the cells out by hand, put them in tubes, and analyzed them by PCR for channel genes. So just here, I think there are about, 15 or so genes measured in single, in this case, PD neurons. And what I want you to see is that there's about a 2 to 6-fold variability of each gene as measured in individual PD cells. There are 2 PD cells in every animal, so this is across animals you have that range. Now this range, that 2 to 6-fold, is the same range, or 2 to 8-fold, whatever it is the same range we see in measurements of synaptic strength, that we see in biophysical measurements of membrane conductances, and it's very similar to the range that many people see in all sorts of other preparations. So this is just showing you that even though the electrophysiological recordings look very, very similar, the conductance densities and the mRNA levels are pretty much the same. And I should say one thing, that even though there's no particular reason why the mRNA levels should match the protein levels, when we have checked in our system, they actually are very highly correlated. And I think that's partially because these are cold-blooded animals and everything is running very slowly. So the protein and the RNA seem to be pretty well matched. So I'd just like to summarize to this point and say individual synaptic and intrinsic parameters can vary 2 to 6-fold across animals, even when their networks have very similar behavior. So next I'm going to show you some results from some conductance-based models. These models are isopotential. They have currents that have been fit to voltage clamp recordings from these neurons. And each of these models has an inward sodium current, 2 calcium currents, a transient and a sustained, an IH, a hyperpolarization activated inward current, 3 potassium currents, a delayed rectifier, that's a conventional delayed rectifier current, a calcium activated potassium current, and a transient outward current, or IA, as well as a leak current. So this model, I like to call these models sort of semi-realistic. They're semi-realistic in that they're computing in conductance, they're grounded in biophysical reality, but obviously they're not capturing all the spatial distribution of the channels or the processes. So they're sort of middle level complexity. Now, I'm going to next show you a couple of slides that deals with this issue of degeneracy. And to do this, I have to show you a way that Leandro Alonso, who's a postdoc in the lab, developed to basically to display well the contribution to each current to the dynamics. At the top, you have a voltage trace of a PD neuron. This is actually, and the PDs are part of a 3-cell pacemaker kernel, and they are capable of producing bursting pacemaker potentials. So here you start seeing at the bottom, you see the cell is hyperpolarized, and then it fires a burst of action potentials, and then it repolarizes. And what Leandro has done to allow you to see what's going on as the interaction, where's the stick? Oh, the stick. Gives you a lot of power. You can see here that what Leandro's done is in black, he's showing you the total outward current, and this is a log scale. Down here in black, the total inward current, and then he's displayed the percentage of the total outward current carried by each of these channels. So in purple, you're seeing the A current. It's pretty small. In orange, the calcium-activated potassium current. In yellow, the delayed rectifier, and you can see it's turning on and off during the spikes, etc. Down here, you can see the sodium current. That's necessary for the spikes up here, the calcium current, the H current, etc. So this just gives you a way of visualizing what's going on. And now I'm using this, I'm showing you this because I wanted to show you the next slide, which is an example of degeneracy. Now this slide looks worse than it is. It actually is very beautiful. You just have to trust me. Okay, there are 6 model neurons here, model A, B, C, D, E, and F. And you notice the voltage traces have very similar waveforms. So if you just ran into them in different animals with your electrode, you'd say that's pretty similar behavior. But Leandro built them with different sets of maximal conductances, that is to say different numbers of the voltage-gated channels, and you'll see that here. If you look in this model, the A current, which is the purple, you can see is tiny. But if you look down here in this model, the A current is enormous. Likewise, the calcium-activated potassium current, which is orange here, is actually quite different than it is there. So even though your Gestalt viewer can tell you that these sets of conductances are opening and closing in quite different dynamics, the overall behavior of the cells is quite similar. So this is exactly what we mean by degeneracy, that every PD neuron doesn't have to be identical, it just has to find a good enough solution. And there's a whole other conversation and a whole other talk that we could have about the homeostatic mechanisms that allow cells to develop these sets of solutions, but be that as it may, you can see they can have quite diverse solutions. Now, I'm actually showing you this slide to show you my favorite slide of that year, which was this one, and this is going to precede coming back to temperature. So what Leandro did is he used a genetic algorithm to find a set of models, all of which had very stable temperature behavior. And you'll come back to that later. And what he's doing now is he's showing you the last spike of the burst in one of those models. And he's showing you at 10 degrees, at 11 degrees, at 12 degrees, all the way up to 25 degrees. And to build these models, he implemented temperature dependence of the activation and inactivation of all of these channels. So he gave the normal voltage-dependent and time-dependent properties of the cells realistic temperature dependence. So that is to say they had Q10s in the 2 to 4-fold range. Okay, but let's look at this now. At 10 degrees, if we look at the purple, we look at the A current, you can see it's pretty big, and the orange, the calcium-active decay, is pretty small. As we go up in temperature, you can see the purple is getting smaller, and the orange is getting bigger, until by the time you look at 25 degrees, the purple is very small and the orange is very big. So this last spike of the burst here and the last spike in the burst here have slightly different waveforms, but more importantly, they have a very, very different configuration of channel opening and closing. Now, the reason I love this slide is because you see a completely smooth transition in mechanism as you go through this temperature swing. And this is really, really important. You probably all know that most neurons in the brain, in your brain, have multiple potassium currents, multiple calcium currents, a whole variety of ion channels. And I like to think that the reason you have so many different kinds of calcium channels and potassium channels, et cetera, is so that you can actually do smooth transitions between behaviors under different circumstances, whether it be temperature or modulation, et cetera, or just frequency. But this allows the neuron to smoothly segue between mechanisms. Now, I'd like to make one more point, especially for those of you who care about disease. If you had gone in to an animal that was operating with this mechanism and blocked the A current, you might not have seen very much. If you'd gone into an animal under these conditions and blocked the A current, you might have seen a much bigger effect. okay? Because here the acorn is doing a lot, and there the acorn is doing very little. And obviously the opposite is true for the calcium-activated potassium current. So even a simple experiment where you say, I'm going to block current this, or optogenetically activate that, or whatever, can have a very different result depending on the conditions in which you're asking that question. And so it's not surprising that people get as variable and as unreasonable effects when they try and understand the mechanisms by which brains work. Okay, I'll skip that. So when we started talking about degenerate, oh, maybe I'll go back and show it. I started talking about degenerate circuits in about 2004 when Astrid Prince was a postdoc in the lab. And this is from a very well-known paper that Astrid produced, where she produced many models of the pyloric rhythm. Here's model network 1, here's model network 2. These are the parameters that give rise to this behavior. These are the parameters that give rise to this behavior. So here you can see at the level of the circuits that this set of parameters is very different from this set of parameters, but nonetheless, they're both able to work well enough. Now, and in particular, if you just look, you can see the H current and the PY is giant here, it's small here, etc. The AB to LP synapse is big here, it's small there, etc. Now, when I first started showing these data, there were some smart physicists in every audience, and they all said, well, I invariably got the comment, oh, this can't be true because everybody knows that biology is always optimal. And so this can't be true. So I said, well, you're not optimal, so biology isn't always optimal. And that usually finished that conversation. It was really rude of me, wasn't it? But it was rude of them to say it couldn't be right when I was showing him. Okay. So, but what they did say, and it's really true, and this was sometimes when people say things, when you hear it 3 or 4 times, you start thinking. So they said, even though this set of parameters and this set of parameters are capable of producing the same thing in the unperturbed case, if you were to perturb these 2 networks, because their parameters are so different, they would definitely respond to perturbations differently. And that's true. And so then they said, well, that's why they said this can't be right. And I said to them, if I have 2 crabs, and one of them has this solution set, and one has that solution set, the question is whether those 2 crabs can respond reliably and robustly to the perturbations the animals are going to see in their life, right? There will always be a perturbation that will separate them. But the question is whether there are the perturbations that they need to deal with, whether they can be still consistent with good enough behavior. So, so we started thinking about this, and so I started thinking about what kinds of perturbations would be useful as probes to try and understand the differences in degenerate neurons and circuits. And the global perturbations that we've been studying since then include temperature, high potassium, pH, and salinity. And we have studied each of these both in the intact animal, in the dissected stomatic gastric nervous system, and under a variety of conditions. And what we're doing, most of what we've studied is temperature and high potassium. We have some really interesting results with high potassium and some really interesting results with temperature. But in all cases, the idea was to look for perturbations that can influence every element of the circuit, because that's what's going to happen if you have a crab living out there in the Atlantic Ocean, or God knows what's happening to the shrimp down in Louisiana now with the cold that's hitting that part of the world. So I'm going to very quickly show you some experiments with the effects of high potassium. And these are experiments that Lily Heed did when she was an undergraduate in my lab. She did a beautiful undergraduate thesis. She went off to Harvard. She got her PhD there. And I don't know what she's doing now. She told me and I forgot. She started a postdoc, but then she told me she was doing something else. So I wish I remembered. I think I wasn't really happy that she was leaving her postdoc. I think she was going off to make a lot of money. Okay, so here are recordings from the PD and the LP neuron. It's fine if she goes off to earn a lot of money, but she's really, really, really a great scientist. So this is a slow time-based recording of a very compressed. So here's the control. Here's high potassium in green. These recordings here are up here. 3 is up there, 4 is there, 5 is there, 5 is after wash. So what you see is the first thing that happens after you go into high potassium, as you'd expect, the cells depolarize and the network stops being active. But, and that's fine, you can say these cells are going into depolarization block or whatever. And then slowly over time, in sustained high potassium, the cells remain depolarized, but the rhythm comes back. So this is really cool. So here is here. By 4, you can see the rhythm has come back even though both cells are quite depolarized. And then when you wash out the potassium, they hyperpolarize back down. So the next thing that we learned, so that's also, first it's interesting because you have this adaptation to the high potassium. And then the next thing that Marder discovered when she did her thesis is that, and here's now 3 applications of high potassium. Here's the first one with silence, and you can see that up on top, silence, and then the rhythm coming back. But what happens is in the second application, the silence is either much truncated or completely gone, and the third, in this case, it's completely gone. So that's telling you is that there's an adaptation that actually lasts for quite a while in between these multiple applications. And so we can call this a memory of sorts that persists over a fairly long period during which the cell has been hyperpolarized and repolarized. And that memory lasts for at least 6 hours. And so it's actually a really interesting perturbation because that perturbation is a short-term perturbation. Eventually it washes out, but it's a sort of an interesting time period, again, about the 6 hours. Now, the reason why I find this so interesting is because the memory, if you want to call it, is cryptic or not visible in control conditions. So if we just look at animal 2 over here, these are recordings from 3 animals. This is the baseline recording. This is the wash when it went back to normal saline after the first application, after the second application, after the third application. The wash recordings, the control recordings, look identical. You have no idea that something's happened, that this long time course memory has happened. That memory is cryptic or hidden until you reveal it by a subsequent perturbation. And I like to think of this as sort of in a way similar to the kinds of things that happen with PTSD, where you can have an event which changes the nervous system, but isn't revealed until you do the appropriate perturbation. Now, the very cool thing that Mara figured out eventually, and this was caused by our work on temperature that you'll see in a moment, Lily had seen, and Mara had seen that sometimes the silent period was very long, sometimes it was medium, and sometimes it was very short. And Mara went back to all the notebooks, and she plotted the time of silence in high potassium as a function of time of year. And you can see here that the short silent periods were in these months, And the long silent periods were in these months. So we have an effect, probably almost certainly, of the water temperature when the animals were fished, that again is completely cryptic, but is only revealed by that perturbation. Okay, now I'm going to skip and go through temperature. But before I do that, I'd just like to show you a very simple model. This was built by Max Kengott, who's a graduate student of mine. And in this case, it's a model with only two currents, a calcium current and a potassium current. But each of these currents has Q10s, or temperature dependencies, associated with their activation and inactivation curves. Okay, in this case, you have this model, Morris-Lacar model, and you see at 22 degrees, the model is pretty similar in the two cases. So here, they're very similar. However, when the Q10 of the potassium current is 2, and I actually think this is the conductance, and the Q10 of the calcium current was 3, you can see as you go colder and colder, the model gets slower and eventually crashes down, and here it's crashed up. However, with different sets of Q10s, now in this case the potassium Q10 is 3 and the calcium Q10 is 2, the model is actually more robust to extreme temperatures. It goes faster at 27 degrees. Here it was crashed, here it's still oscillating, and down at 7 degrees, it's giving you a slow oscillation, and here it was silent. So this is just showing you that the same maximal conductances, but with different temperature dependencies, can give you different phenotypes. Okay, so the pyloric rhythm. That triphasic rhythm you saw before, here with extracellular recordings, these are data from Lamont Tang. At 7 degrees, you have a slow rhythm. At 19, you have a faster rhythm. And however, the phase relationship says that the normalized time at which the cells are active and inactive are completely constant over those temperature range. And that's actually remarkable. However, if you go to higher temperatures, and this is different for every preparation, you get what we call crashes. And this is when the rhythm becomes disrupted. And these are just 4 different crashes that Lamont recorded many years ago in 4 different experiments. All of them were normal under control conditions and indistinguishable, but they all crashed with different kinds of complex dynamics. And that's exactly what you'd expect if they start out with different underlying sets of conductances as they respond differentially to the different perturbations. And then what I would just like to show you is 1 or 2 slides from the model that Leandro built. So he built a bunch of models, all of which were well temperature compensated from 10 degrees to 25 degrees, and then crashed at 30 degrees. And to do this, he used genetic algorithms to find these successful solutions. And you can see 10, 15, 20, 25. The triphasic rhythm is there. The phase relationships are maintained, but the frequency goes up. So that's just exactly what happens in biology, and then the crash. But I'd like to show you, the reason I'm showing you this now is to show you this slide. This is a little more complicated. So in this case, on top, you have a circuit where everything is intact, and it's at 10 degrees. And then what Leandro did is he turned off the A current at 10 degrees, and you can see the rhythm is completely disrupted. So this is a case where the A current was completely necessary for the generation of this normal rhythm at 10 degrees. However, when he took the same network to 25 degrees, the network went faster, as he'd predict. But now, when he turned off the A current, you can see it's slightly disrupted a little bit, but basically it's fine. So in this case, the blockade of the A current was very, very, very pernicious at 10 degrees and had almost no effect at 25 degrees. In contrast, here's a case where he manipulated the calcium-activated potassium current. Here you have at 10 degrees, the normal rhythm. He turned off the calcium-activated potassium current here, and you can see almost the rhythm is persisting. It's slightly different frequency, but basically it's doing fine. But when he did the experiment at 25 degrees, here you have a faster rhythm because it's at higher temperature. And now when he turned off the calcium-activated K, the rhythm is completely crashed. It's completely gone. So again, this tells you or shows you that the effect of the perturbation is very temperature dependent. In this case, it's worse at 10 degrees, and in this case, it's worse at 25 degrees. And that gives you some sort of insight into why the human nervous system can be so differentially affected by temperature, especially if people have different kinds of neurological disorders with different kinds of circuit and cellular configurations. Okay, now the last little story. This goes back to 2012. Sarah Haddad was just starting her graduate work in my lab, and she wanted to repeat Lamont's data, and I always like it when people start out trying to repeat what somebody else has done. So I told her just to do a temperature ramp, and she came back and she said they're not crashing. I said, of course they're going to crash. And I said, you're doing something wrong. You don't have the thermometer in the right place. And she got really angry at me. And she came back and she said, I put 2 thermometers in, and you're wrong because at 36 degrees, the rhythm is there, and it's going 7 hertz or 6 hertz. And I said, oh my God, I've never seen a rhythm go that fast. So here we have 11, 20, 25, 30, 36. And then we realized later that 2012, there was a winter that never was. The crab habitat was 6 degrees centigrade warmer than normal. The animals in Woods Hole, the squid went to Maine, the dogfish went to Maine, our crabs couldn't migrate that much, but we had animals that were way, way, way too resilient to high temperature. Then the next year, the crash temperatures came back down. We were sort of hoping, okay, maybe life has gone back to normal. And then in 2021, Mara got these data. 11, 15, 19, 23, 27, 31, 35. You can see the rhythms were maintained over a very large range of frequency again. And then what Mara did is something that made me very proud of her. She went back to our lab notebooks and she mined the data for recordings from 2008 to her recordings then it was 2021. And so these are recordings from Lamont in 2006. Here's 11, 21, and 31 degrees. It was completely crashed at 31, perfectly fine at 11. 2012, these are some of Sarah's data. 11, 21, 31, 35. These were Jacob Ratliff's data from 2016. 11, 21, 31 completely crashed, a lot like Lamont's data. And here are some of Mara's data from 2021, 11, 21, 31, 39. And then Mara went one step further, and she went to the NOAA website, and there's a buoy 16 miles off the coast of Boston, and she picked the surface temperatures off that buoy, and at Lamont's year, these were the summer temperatures. Here's his data. Here's Mara's data in 2021. And you can see there's a correlation between where the crabs were crashing and the water temperature. And these are the winter temperatures. And so here you can see the crash temperature as a function, crash frequency as a function of temperature. Here's the crash temperature in these different years, 2016, 2020. And you can see the water temperature that the animals were living in greatly influences their subsequent crash temperature. So what made me really, really sort of distressed by this well, I was really happy that Marder was able to find all the data in the lab. That was good. And I highlighted this, and you can tell I highlighted it because it's a real mess, right? I'm not so great with PowerPoint. But if you look at the 11-degree data, all of those 11-degree data look normal. 11 degrees is our control data set. So if we hadn't done these perturbations, we never would have known that the animals were different, because at 11 degrees they all look the same. So hidden in all of the experiments that we all do, there were the effects of the temperature experience of the animals that were completely, again, completely cryptic, because we weren't doing the perturbation that was going to show that. So now, of course, we keep incredibly good records for our tank temperatures. We're doing a whole series of acclimation experiments, but it's all about trying to really pin down the time course and the results of different temperature experience on the subsequent effects of the preparations to perturbations. So I'd just like to end by saying that degenerate circuits can hide cryptic differences due to differential history that can alter or shape future responses to additional challenges. And this is the challenge to us, because no matter how well you think you're controlling the development of your animals, even if you're doing it in an animal room, there are many things that you don't know what's going on. And in the case when you're working in wild-caught animals, they have a whole life before you get them. And so what we're trying to do now is to control some of that, to really try and understand better the molecular and cellular mechanisms by which, in this case, long-term temperature experience changes the responses of the animals to the perturbations that they see in their life. And I'll stop there. Do I get to, Kelsey?
All right, I'll remind everybody when they ask a question to use the microphone at the back of their seat. But so maybe, I'm thinking about the experiments you showed where there was this 6-hour sort of memory. And what you're showing that, the data showing the temperature in the ocean, I guess near Woods Hole, or wherever they're caught.
Boston Harbor.
Harbor.
These are our animals.
But you keep them in tanks, and I assume the tank is at a set temperature. So does it mean that there's an even longer term?
So acclimation. In the acclimation literature, in the temperature literature, people will tell you that temperature acclimation takes 3 to 4 weeks at least. I skipped some experiments that Lamont did where it showed that 3 to 4 weeks gives you some amount of acclimation, but not as much acclimation as we saw in these 4 and 5 month periods in the ocean. So what we do now is we have animals in tanks at 11 degrees, that's the control. We also have animals at 7 degrees and at 19 degrees, so we're acclimating, and we are trying to keep them for at least 4 weeks. And then we're going to study the effects of acclimation on acute temperature, potassium, modulation, and all sorts of other things to try and understand that. Yes, that 6-hour potassium is a really interesting time constant in the brain. The temperature acclimation and many other features probably are slower still. So I think so. Now, the experiment we want to do, which I probably won't be able to do if the NIH shuts down, okay? If the grant that may come through, which may not ever come through, is we want to set up a scenario where we do cyclical changes in temperature, because in the ocean, in our tanks, it's easier just to hold them at 11 constantly or 19 constantly, but that's not the experience they have in the ocean. And I suspect that the more dramatic effects we get from the ocean acclimated animals are because they're being challenged. So there's some experiments in insects which say you get more effective acclimation if you challenge them and come back down and then challenge them again. It's like weight training of your muscles. If you want to get stronger muscle, doing it every day or then resting and then doing it again is more effective than just holding, right? So yeah, but So probably to do the cyclical experiments, we might have to go to a marine lab. Yeah.
Thanks, great talk. So I was wondering, so if there's all this, even better. Thanks. Hi. So if there's all this degeneracy with these different channels and all, so is there like a master controller that figures out how all these should come together to result in a particular behavior every time? How does that work? Like, how do they all know, how to?
So the talk I didn't give is about that. Okay. So for many years, I started with Larry Abbott in 1993. We started building models that would homeostatically allow you to build and maintain a certain activity pattern. The way these models all work, and the most recent one was just published, the way these models all work is they say that you can use intracellular calcium concentrations as a way of the cell being able to monitor its own activity. And then if you have calcium-dependent processes that control either transcription, translation, or other kinds of modifications, you can then, by setting a sort of a calcium target, you can basically let a cell build itself to a certain activity profile. So the idea would be every cell would have a way of setting its preferred activity profile, and it would do that by modifying the properties of a lot of the ion channels in its membrane. And these models work. And there's actually a lot of experimental data that suggests these are right too.
Do we know what that memory mechanism is, how that works?
In the high potassium?
Yeah.
No. No, we know that it's a long enough period of time to start activating heat shock proteins and a whole variety. So there are a whole bunch of molecular things get triggered by the high potassium as well as by temperature as well as by all these other perturbations. But we don't know about the high potassium one. We know a lot of things it isn't, but we don't actually know. That's the downside of working with wild type invertebrates. Yeah, there's a question back there that I can't see because it's hidden in the dark.
I have a question. So when you catch these crabs, all the currents were different, although the circuit behaved the same.
Right.
Thank you. So do you think that mostly reflects the life history of those crabs, or do you think that this is like some genetic variability in the population?
Both.
And
Wouldn't you think both?
And so when you catch these crabs on those hot years, then the guys that grew up or were captured or exposed to the hot years, those guys seem to be more, their circuit still work even when it was still oscillating, even it was really hot, right? Like 36, right? So do you think that reflects adaptation, or do you think is that the guys that could not survive, you didn't capture and they die?
So we don't know. I mean, that we should be able to figure out using long-term acclimation experiments. So what we do know is that if you try and keep animals very warm for a long time, some of them die and some don't. In lobsters, we're doing some stuff with lobsters now. The hot animals become really, really aggressive and nasty. So there's actually a really interesting change in behavior associated with long-term acclimation. We're assuming in the case of the crabs that there are probably some animals that died because they couldn't handle the temperature, and others just went through this acclimation process. And obviously, we have no way of knowing because the fishermen are pulling in what they're pulling in. But if we do the acclimations in the lab, we should be able to figure it out.
Is there a question over there?
Was there a question over here? See, that's what's wrong with this room. You can't.
Is this on?
The room is very pretty.
Wait, should I wait for the mic, or this sounds good? Okay, I've got it. So great talk. I'm very excited about the oscillation story, and I want to see where it goes. Could you tell us a little bit about what you saw with pH?
I can't hear you.
Oh, you couldn't hear me? Okay. One more time. Really excited about seeing where the oscillation story goes, if it's circuit level or not. It sounded like
It
temperature.
is circular. Well, it's both.
Like the synaptic currents, do we know how much they contribute? Is one question then.
Yes,
And
we've known for 40 years.
okay, so what? Okay, sorry.
Okay.
Well, I'll come back. But the other question would be, what is the pH effect that you ended up seeing?
So pH is very interesting. Unlike us,
Yeah.
we tend to be our nervous systems are pretty pH sensitive. We did experiments where we took the pH up to 10 and 11 and down to 4 and 6. And we saw almost no effects of pH until we got above 10, above 9, and below 6. So these animals are able to deal with a much bigger pH range than we can. And probably that's because they live in the ocean, which is a bicarbonate buffer. And when they go up and down in the water column, the amount of dissolved oxygen, dissolved CO2, and therefore the pH, as well as the temperature, changes. So they are built to be far more pH and oxygen tolerant than we are. So what we've given up this environmental resilience in order to go fast. We've done a bargain with the devil. We run hot because we wanted to run fast.
Well, thank you. So over evolution, DNA has implemented our billions of molecules and cells, right? So over evolution, DNA has implemented this effect of temperature, right, in nature, right? Because temperature they have changed, and this is adaptive. The DNA has to be adaptive in terms of implementing this effect of temperature and
So there's a little nasty lesson, which is lobsters and crabs have a very complex genome, and there's a ton of non-coding stuff in there, and in octopus, and there is a whole lot of RNA editing, and so there's probably the case that in these long-term acclimations that you see in the ocean, there may be some effects of RNA editing and circular RNAs and all sorts of other things hidden in that, so that the genes for the sodium current are still there, they're still the same, but there may be modifications of various and sundry things that we don't know that are actually produced from all this non-coding DNA that helps in these long-term acclimations. And that's what we suspect.
Alrighty, well, join me in thanking Dr. Marder for a brilliant talk.